Sathyabama Institute of Science and Technology.
Dear students, this is Dr. S.M. Indhumathi from the Department of Biotechnology, School of Bio and Chemical Engineering, Sathyabama Institute of Science and Technology. Today I am here for a presentation on the topic Mechanism of Prokaryotic Transcription. Let's first have a glimpse of what is transcription. The central dogma of molecular biology implies that DNA is copied to make mRNA through the process called transcription, while the mRNA are in turn translated to produce proteins. The process of transcription is basically the first step of gene expression, where DNA tries to express itself to form the final product called proteins. Transcription occurs in both prokaryotes as well as eukaryotes in a relatively more or less similar fashion. In transcription, one of the two strands of DNA called the antisense strand or the non-coding strand actually serves as a template to make mRNA with the help of the enzyme RNA polymerase II. Transcription is basically the most important step in any living organism because it makes proteins. So proteins, they make up the cells and tissues of all the living beings. So if transcription is inhibited or blocked through any of the inhibitors like alpha amanitin, that's a mushroom toxin, or any other antibiotics like rifampicin or actinomycin like that, if through this process, if transcription is blocked, the formation of proteins will be affected. If proteins are affected, that will lead obviously to cell death. And RNA polymerase becomes a crucial enzyme because that carries out this transcription in prokaryotes, especially RNA polymerase II carries out the transcription process here. And the site from which DNA gets transcribed is called as the transcription start site, and that is called, the nucleotides present from that transcription start site are given positive numbers and they are called downstream nucleotides, and those nucleotides that are present before the transcription initiation site are given negative numbers and they are called upstream nucleotides, and the promoter region lies in this particular upstream region of your DNA strand. And if the gene that has been transcribed encodes a protein, that will be finally translated to make a chain of polypeptide, finally ending up with the product called proteins. And transcription follows three major steps, that is the initiation, elongation and termination. Before we get to know what is initiation, we must know what promoters are. Promoters are longer sequences or stretch of nucleotides that are present upstream of a DNA strand. As I've said earlier, upstream nucleotides are present before the transcription initiation site, and all the nucleotides are given negative numbers, and those nucleotides that follow the transcription initiation site afterwards are given positive numbers, and they are called downstream nucleotides. This promoter region actually enables or it makes the RNA polymerase to know where to exactly sit down on the DNA strand and initiate the process of transcription. And promoters also ensure the direction of transcription, that is, the transcription always follows in 5' 3' direction. So coming to this initiation process, in the promoter sequence, we have two particular elements called the -10 element and the -35 elements, we say. Why they are called -10 and -35? It is because the -10 elements are located 10 nucleotides far from the transcription initiation site, and the -35 elements are located 35 elements from the transcription initiation site. The RNA polymerase selectively recognizes these two elements and attaches itself to it, marks the initiation of transcription. So after the binding of RNA polymerase II selectively to these two elements, the -10 element as well as the -35 element, this circle in the image resembles, it indicates RNA polymerase II. It has bound to the two elements -10 and -35 here, and after binding, the RNA polymerase immediately starts to unwind the double helix of the DNA strand, making the transcription bubble to open. And this unwinding of the DNA strand occurs immediately after the stretch of this AT sequences, that is the -10 elements. Why unwinding of DNA happens immediately after this minus 10 elements means because there are continuous stretch of 80 nucleotides in both the strands. The interaction between these two nucleotides in both the strands will be more or less weaker because of the presence of double hydrogen bonds. So it will be easy for an enzyme to break the double hydrogen bonds present between these two strands, while it will be difficult for an enzyme to break the triple hydrogen bonds that are present between the GC nucleotides. So immediately after binding to these two elements, the RNA polymerase starts to unwind the double helix of the DNA strand, thereby opening the transcription bubble, and this portion is called the transcription bubble. You can see where this is the minus 10 element. After the 10 element, immediately the RNA polymerase unwinds the DNA double helix and makes the transcription bubble open. It looks like a balloon form in the DNA strand. So this is our 5' 3' strand, which is called as the coding strand or the sense strand, which does not serve as the template here, while this strand 3' 5' strand, which is the anti-sense strand or the non-coding strand we say, that strand serves as a template on which you have the promoter region, which has in turn minus 10 and minus 35 elements to which the RNA polymerase selectively recognizes and binds and initiates the process of transcription by opening the transcription bubble. So this process marks the initiation of transcription. Following initiation, we have the elongation process. Elongation process is the one during which the RNA polymerase walks along the DNA strand, reads the nucleotides and tries to add the complementary nucleotides to the growing mRNA strand. As I have already said, transcription always follows this 5' 3' direction. RNA polymerase 2 tries to add, it adds nucleotides, different nucleotides, that is the complementary nucleotides to the 3' end of the growing strand of mRNA. The growing strand of mRNA now resembles the coding strand of the parent DNA. That is, the 5' 3' strand will be exactly similar to that of the growing mRNA now, except for the presence of uracils, because mRNA will be having uracils while DNA will be having thymines. This process of elongation will continue as long as the mRNA meets a stop signal or a termination codon. And finally, elongation ends up with termination of transcription. And as you can see, so this is the RNA polymerase that is bound to the promoter region that is present in the antisense strand, and now your RNA polymerase tries to add complementary nucleotides to the 3' end of the growing mRNA strand, and this strand will be exactly similar to that of this template, parent strand, which has this 5' 3' regions. Then comes termination. So in the process of termination, when your mRNA meets a stop signal or a stop codon or a termination point or a termination sequence, the entire process of transcription stops. In prokaryotes, we have two different types of termination called row dependent and row independent termination. Certain prokaryotes have a binding region in their mRNA for the binding of the rho factor. Rho factor is nothing but it is a hexameric helicase. It is actually a protein factor. So this rho factor binds to the mRNA and stops transcription in row dependent termination. So in row dependent termination, what happens is, so this is the binding site for the rho factor in the growing mRNA strand, and the rho factor has identified that binding region and has gone to bind with it. After the binding of the rho factor with the growing mRNA strand, the rho factor starts to travel upwards to reach the growing mRNA strand. Once the rho factor reaches the transcription bubble and when it meets the RNA polymerase there, it immediately stops the process of transcription, thereby putting an end to it. It pulls off the growing mRNA from the transcription bubble and the entire process of transcription stops here. But in case of row independent termination, I have already said some prokaryotes do not have binding region for this rho factor. In such prokaryotes, intrinsic type of termination or row independent termination occurs. So this is row independent termination, where there is no requirement of particular stop signal or termination sequence or something like that. Here there is no binding of rho factor. So what happens is, the growing strand of mRNA, when they hit a region that is rich in GC nucleotides, rather than growing in a linear form, the mRNA will actually fold back itself to make a hairpin or stem loop like structure like this. And the successive GC nucleotides, that is the forthcoming GC nucleotides, will actually become complementary nucleotides when the mRNA folds back to itself. So, this is a continuous stretch of GC nucleotides here. So, the formation of this hairpin loop actually stops the activity of RNA polymerase. Actually, it slows down the activity of RNA polymerase. Finally, it halts the effect or the activity of RNA polymerase, and the end of this transcribed mRNA will be actually rich in U nucleotides. If there are continuous stretch of U nucleotides in mRNA, it implies that there are continuous stretch of A nucleotides in the template DNA. So, the interaction between the A nucleotides and the U nucleotides will be very much weaker. So, the formation of this hairpin loop as well as the weaker interaction between the A nucleotides and the U nucleotides completely stops the transcription, and thereby it will pull off the primary transcript of mRNA outwards of the transcription bubble. The mRNA is now released and RNA polymerase actually falls off from the transcription bubble, thereby putting an end to the transcription process. So, this is all about the process of prokaryotic transcription. These are my references. Thank you students for patiently watching the presentation.